Biochemistry 401, Lecture 21. Today we're going to talk about the electron transport chain. And to do this, we're going to have to talk about the structure of the mitochondrion first. Then the transport of reducing equivalents from the cytosol into the mitochondrial matrix. We're then going to have an oxidative phosphorylation overview, and then we're going to talk about the electron transport chain in particular. And that consists of integral membrane proteins that are electron carriers and also mobile electron carriers. We're going to finish with following electrons through the electron transport chain. So let's get started. Before we go any further, we're going to take a look at mitochondrial structure. We're going to walk from the outside in, beginning with the outer mitochondrial membrane that is shown in dark brown and the text box shown in orange. The outer mitochondrial membrane is a freely permeable membrane, and most small proteins, metabolites, and ions can go through this membrane. And so, in many ways, it's almost continuous with the cytosol. This is because there are porin proteins that are found in this outer mitochondrial membrane that allow such molecules to freely go back and forth according to their concentration gradients. Now, walking from the outside in, the next thing we're going to hit is the intermembrane space that lies between the outer mitochondrial membrane and the inner mitochondrial membrane. This is going to play an important role. The next thing we're going to see is the inner mitochondrial membrane. This is a highly impermeable membrane, and most things can't get through it unless there is a transporter that transports these small molecules or proteins through this membrane. This inner mitochondrial membrane contains the electron transport chain, and it also contains ATP synthase. This inner membrane is thrown into a series of folds called cristae. The role of these cristae is to increase the surface area of the inner mitochondrial membrane to increase the number of enzymes that are found there. It's recently been shown also that ATP synthase is located at the top of each of these folds in a double row that helps to stabilize the cristae in this shelf-like form. Pretty cool stuff, actually. Continuing on our walk, the next thing we're going to see is the mitochondrial matrix. This is the central interior portion of the mitochondrion that contains the circular genomic DNA, some RNA and ribosomes, pyruvate dehydrogenase complex that we saw. We also saw the citric acid cycle. We're going to see some fatty acid oxidation and amino acid oxidation. And as we know, there are some reactions involved in heme synthesis that are found here as well. And as we'll see, there are some reactions that occur in the mitochondrial matrix that are involved in urea synthesis. So some reducing equivalents are produced in the cytoplasm, in cytoplasmic pathways like glycolysis. But the electron transport chain happens in the mitochondrion. So we have to find a way to get those electrons into the electron transport chain in the mitochondrion. But we said that the inner mitochondrial membrane is highly impermeable. And the thing is, there's no direct route for reducing equivalents like NADH to pass through this inner mitochondrial membrane. There's no transporter for this molecule. So what happens is there are two main shuttles that transport electrons from the cytosol into the mitochondrial matrix. The first one is the glycerol 3-phosphate shuttle, and the second one is the malate aspartate shuttle. We're going to start with the glycerol 3-phosphate shuttle because that's much more simple than the malate aspartate shuttle. The glycerol 3-phosphate shuttle uses cytoplasmic NADH and mitochondrial FAD. Now this mitochondrial FAD is shown here as EFAD because this FAD is actually a prosthetic group that's bound to an enzyme, and we'll look at this in a little while. With the malate aspartate shuttle, we begin with NADH carrying the electrons in the cytoplasm, and we're going to pass those over to an NAD plus molecule that's in the mitochondrion. We end up with NAD plus in the cytoplasm and NADH in the mitochondria. This is a glycerol 3-phosphate shuttle. In this process, we're going to start with NADH that's in the cytosol, and we're first going to pass those electrons to dihydroxyacetone phosphate to reduce it to glycerol 3-phosphate. We're then going to use an enzyme associated with the inner mitochondrial membrane. It sits on the cytoplasmic face of the inner mitochondrial membrane, facing the intermembrane space. Now this process is going to happen in 3 steps. The first thing we're going to do is we're going to reduce dihydroxyacetone phosphate to yield glycerol 3-phosphate. And so the reaction is dihydroxyacetone phosphate plus NADH plus a proton yields NAD plus glycerol 3-phosphate. Dihydroxyacetone phosphate is being reduced, and NADH is being oxidized to NAD plus. Now these electrons from glycerol 3-phosphate can be handed off to another electron carrier. This is bound to an enzyme, the mitochondrial glycerol 3-phosphate dehydrogenase. We're going to reduce FAD that's bound in this enzyme to yield FADH2, and in the process, glycerol 3-phosphate will be oxidized to form dihydroxyacetone phosphate again. But this shuttle doesn't stop here. The electrons that are carried by FADH2 can be passed off to a mobile inner mitochondrial membrane electron carrier that is known as ubiquinone. Now, ubiquinone is sometimes called Q10. This is a mobile carrier that is lipophilic and shuttles back and forth in the inner mitochondrial membrane, carrying electrons from one electron carrier to another. Ubiquinone is often abbreviated Q. This is the oxidized form of the electron carrier, whereas QH2, ubiquinol, is the reduced form. We will look at this more closely in a little while. This is the malate aspartate shuttle. It is much more complex than the one we just looked at, and we're going to follow this really slowly so that you can figure out what's going on. The enzymes that are involved in this are a malate dehydrogenase in the cytoplasm, an aspartate aminotransferase in the cytoplasm, a malate dehydrogenase in the mitochondrion, and an aspartate aminotransferase in the mitochondrion. Now, both of these mitochondrial enzymes are in the mitochondrial matrix. And so we're going to start off with oxaloacetate in the cytoplasm, and we're going to reduce that to form malate. As we do this, the electrons from NADH will be transferred to oxaloacetate to yield malate and NAD+. So oxaloacetate is reduced to malate, and NADH is oxidized to NAD+. Malate will leave the cytoplasm and go into the mitochondrial matrix. Once there, it will be oxidized again to form oxaloacetate and reduce NAD+ to NADH. And so now we have our electrons transferred from the cytoplasm into the mitochondrial matrix. And that's what we want. However, what we don't want is we don't want a lot of oxaloacetate in the mitochondrial matrix, because this might slow up the TCA cycle. And so we're going to move this oxaloacetate out of the mitochondrial matrix. But wait a minute, we can't do that because we can't transport oxaloacetate directly out of the mitochondrial matrix. There's no transporter. So we're going to use another one of our tricks, and we're going to add an amino group to oxaloacetate. Now remember, at the beginning of the semester I said it's important to know which amino acids have 4 carbons and which have 5. And I said it's important to know which metabolites have 4 carbons and which have 5. And this is why. Oxaloacetate has 4 carbons. So does aspartate. And so now what we're going to do is we're going to take an amino group from glutamate and transfer it to oxaloacetate. When this happens, oxaloacetate will be changed to aspartate. There are 4 carbons in oxaloacetate and 4 in aspartate. Aspartate can now leave the mitochondrial matrix by way of a transporter. But let's go back to the mitochondrial matrix for a minute. We took the amino group off of glutamate. This is the alpha amino group. And when we do this, we change glutamate into alpha ketoglutarate. 5 carbons in glutamate, 5 carbons in alpha ketoglutarate. This is the same intermediate that we saw in the TCA cycle. So we don't want this to build up either. And what we're going to do is we're going to transport this out of the mitochondrial matrix by way of a transporter. Once this alpha-ketoglutarate is in the cytoplasm, it can accept an amino group from aspartate to be turned back into glutamate to begin the cycle again. And aspartate, once it gives off that alpha amino group, is changed from the 4-carbon aspartate into the 4-carbon keto acid, oxaloacetate. And in this way, this cycle is perpetuated. Take a minute to look at the arrows, and please note that when you see the transamination, what we're really doing is we're crisscrossing those arrows. So an amino group is going from glutamate to oxaloacetate, and oxaloacetate is becoming aspartate, and glutamate is becoming alpha-ketoglutarate. And just to make it a little more clear, let's look at the amino transfer that's happening in the matrix. We'll start off with oxaloacetate. It is going to pick up an amino group from glutamate to yield aspartate. So oxaloacetate plus an amino group yields aspartate. Glutamate minus an amino group is going to yield alpha-ketoglutarate. And you can see the crossing of the arrows to represent where glutamate is going and where oxaloacetate is going. Aspartate aminotransferase works by way of an ordered bimolecular reaction. And what this means is these reactants engage with the enzyme in an ordered manner. First, aspartate is going to come in and is going to lose an amino group. This amino group is going to become attached covalently to the enzyme itself. And in so doing, we're going to be left with oxaloacetate and an amino group that's covalently linked to the enzyme. Oxaloacetate leaves, and now in comes the second substrate, alpha-ketoglutarate. Alpha-ketoglutarate comes in, and it accepts the amino group from the enzyme. This causes alpha-ketoglutarate to become glutamate, and glutamate leaves. Both oxaloacetate and alpha-ketoglutarate are alpha-keto acids. But what does this mean? This is an alpha-keto acid that has 3 carbons. It's an alpha-keto acid because the ketone is in the alpha position relative to the carboxylic acid that is shown here. And so this is an alpha-keto acid. Likewise, a beta-keto acid has the ketone group at the beta position, and a gamma-keto acid has the ketone at the gamma position, and these are all relative to the carboxylic acid. You should know the identity of each of these alpha-keto acids. Can you identify them? Well, the 3-carbon alpha-keto acid is pyruvate. The 4-carbon alpha-keto acid is oxaloacetate. And finally, the 5-carbon alpha-keto acid is alpha-ketoglutarate. You should be able to identify these structures. This is going to be important now and also later in the semester. And so, reducing equivalents like NADH and FADH2 are produced in such pathways as glycolysis, fatty acid catabolism, the TCA cycle. These are all oxidations of fuels. And now that we have these reducing equivalents, what are we going to do with them? Well, these 2 electron carriers have high electron transfer potential. These electrons can be transferred from carriers of high electron potential to those of lower and lower and lower electron transfer potential. And in so doing, as these electrons are passed from carrier to carrier to carrier, energy is released. And so, as these electrons are passed on through the electron transport chain, energy is produced that can do work. This work is going to be the pumping of protons from the mitochondrial matrix into the mitochondrial intermembrane space. This is going to set up a chemiosmotic gradient that will help to power ATP synthesis. And so what is electron transfer potential? Well, this is something that can be quantified through the use of the expression delta E prime sub zero. This refers to a molecule's reduction potential. And so we can relate reduction potential to the change in free energy under standard conditions at pH 7, because we are biochemists after all. And we come up with this particular equation, where N is the number of electrons and F is Faraday's constant, which is 96.48 kilojoules per mole per volt. Here's a table which is similar to that which we saw when we looked at phosphoryl transfer potentials. Please do not memorize this slide. You do not have to memorize a single thing on it. All you need to know is that the reductant, NADH, has a higher electron transfer potential than does water. And you also need to know the more negative the reduction potential, the greater the electron transfer potential is for the reductant, and the greater the tendency is to donate electrons. The more positive, the greater the tendency to accept electrons. And so let's look at this table, especially let's look at NAD plus and NADH, and oxygen and water. NAD plus and NADH are the oxidant-reductant pair, and NADH has a reduction potential of negative 0.32 volts. This means that NADH is a potent reductant and will reduce anything that is lower on this table. And oxygen is at the very bottom of this table. It has an electron transfer potential of positive 0.82 volts, and so it has more of a tendency to be reduced. So for every redox reaction, you have a reactant that's going to be oxidized and one that's going to be reduced. In this whole electron transport chain, we're going to start off with NADH that's reduced, and we're going to end up with oxygen that is reduced to yield water. So we're going to transfer those electrons from NADH eventually to oxygen to produce water. So the first half reaction that we see here is going to be reaction number 2. This is 1/2 O2 plus 2 protons plus 2 electrons yields water. And the electron transfer potential for this half reaction is a positive 0.82 volts. The next half reaction that we have to look at is reaction number 3. This is NAD plus H plus 2 electrons yields NADH. And the electron transfer potential for that half reaction is negative 0.32 volts. But wait a minute, we're not looking to reduce NAD plus, we're looking to oxidize NADH. And so we have to flip this half reaction, and we get reaction number 4. NADH yields NAD plus a proton plus 2 electrons. And because we flipped the half reaction, we also have to flip the sign. And so this electron transfer potential becomes positive 0.32 volts. I hope you got that. We're then going to plug these individual electron transfer potentials into this equation, and once we get the delta G naught primes, we're going to add them together to see what we have for the sum. So let's do that. So let's plug these numbers in. For reaction number 2, the number of electrons is 2, so negative 2 times Faraday's constant, which is 96.48 kilojoules per mole per volt, times the change in transfer potential. And so that is 0.82 volts. And we get negative 158.23 kilojoules per mole. And now for reaction number 4, the final value is negative 61.74 kilojoules per mole. So we're going to add reactions 2 and 4 together to get negative 219.97 kilojoules per mole. Now that's a lot of energy to release at one time, 219 kilojoules per mole. If we were to release all of that energy at once, some of that energy could be lost to heat or entropy. So, like many things that we do in the body, we transfer this energy in little tiny steps. Let's take a look at that. So how do we use reducing equivalents to get energy for ATP synthesis? Well, in many ways, it's similar to the transduction of potential energy of this beautiful river, the Niagara River, as it's at higher elevation. As this river descends down the falls, this potential energy is transduced into kinetic energy. Now, some of this kinetic energy can be harnessed and transduced into mechanical energy that is then transduced into electrical energy. And this is what happens at a hydroelectric dam that we see here. So how does that relate to what happens in the mitochondrion? So what we have is we have chemical energy in the form of high energy reducing equivalents, NADH and FADH2. The electrons from these electron carriers start up at a high energy and then progressively go to lower and lower energy level electron carriers. And as these electrons are passed from carrier to carrier to carrier, going from electron carriers with high electron transfer potential to lower and lower and lower electron transfer potential. Part of the energy difference is used to pump protons from the mitochondrial matrix across the inner mitochondrial membrane and into the intramembrane space. What this does is it sets up a potential that's similar to water behind the hydroelectric dam. We have a lot of potential energy stored in the intramembrane space as these protons are at high concentration in this region, this small space. And so it's the tendency of these protons to want to flow down their concentration gradient into the mitochondrial matrix again. The release of this chemiosmotic gradient in the intramembrane space is prevented by the inner mitochondrial membrane. We said that it is impermeable, and it certainly is impermeable to protons. They can't just go through that membrane on their own. They have to go through this membrane in a specific manner. And one of these methods is by going through ATP synthase. And as it goes through, it causes a turbine-like motor in the ATP synthase to turn. And in this way, the chemical energy from the chemiosmotic gradient is transduced to mechanical energy. And then this mechanical energy is then transduced to chemical energy again as ATP synthesis happens from ADP and inorganic phosphate. This is a schematic of the electron transport chain, and you need to know this very well. So electrons basically come in from glycerol 3-phosphate shuttle, from NAD8, and from the entry of electrons from succinate via complex 2. And we saw that in the TCA cycle. NADH donates its electrons to complex 1, the glycerol 3-phosphate shuttle, and both complex 1 and complex 2 donate their electrons to something called coenzyme Q. This is a small molecule identified by the letter Q in this diagram. As you can see, Q is found right in the inner mitochondrial membrane, and it travels back and forth in the plane of the membrane. This is because it's a hydrophobic molecule, and so it can do quite well in this lipophilic environment. It shuttles electrons from complex 1, the glycerol 3-phosphate shuttle, and complex 2 to complex 3. From complex 3, electrons are donated to cytochrome C. Cytochrome C is a small protein shown in red here. It is indicated by the letter C in this diagram, and it is shown attached to complex 3. It's going to transport electrons from complex 3 to complex 4, and it travels in the intermembrane space from one complex to another. So it goes from complex 3 to complex 4, and it donates its electrons to complex 4. Now in complex 4, there's oxygen bound, and so complex 4 is going to pass its electrons to oxygen to yield water. Now it's important to note that the electrons that come in via the glycerol 3-phosphate shuttle or complex 2 come in at the level of coenzyme Q. These electrons completely bypass complex 1, and this is an important thing to remember. It's going to have implications concerning the amount of ATP that can be made from these molecules. We're going to look at this a little bit later on. This is our cartoon of the electron transport chain, And in this cartoon, we're going to follow the progression of electrons from one electron carrier to another. And we're going to look at which complexes actually can pump protons into the intramembrane space, because not all of these electron carriers will do that. Each of these large yellow blobs in this cartoon are actually integral membrane protein complexes. So we see complex 1, 2, 3, and 4 are integral membrane proteins. Complexes 1, 3, and 4 all pump protons, but poor little complex 2 does not. Let's look at the progression of electrons through the electron transport chain and look at the electron carriers themselves. NADH will reduce components within complex 1, and in so doing, the energy that's released will be used to pump protons from the mitochondrial matrix into the intermembrane space. This is a yeast complex 1, and the matrix is shown at the bottom, and the intermembrane space is shown at the top of the diagram. This is an X-ray structure of this complex. In the first panel, panel A, each subunit is a different color, and so you can see why this is called a complex. In panel B and C, we see the actual electron carriers that are in this complex. These are iron-sulfur clusters and also FMN that are found in this enzyme. Now, we start with NADH that transfers electrons as a hydride. You'll remember that's 2 electrons and 1 proton. But as these electrons are passed through complex 1, only 1 electron is going to be passed at a time. FMN can pick up 2 electrons and 2 protons, but it's going to give off these electrons one at a time to the iron-sulfur clusters. And these iron-sulfur clusters are going to pass electrons only 1 electron at a time. And this is why we need something like ubiquinone to pick up these electrons at the end of this chain of iron-sulfur clusters. Why? Because we need something that can pick up 1 electron at a time. And so electrons can enter the electron transport chain through complex 1 via NADH, but they can also enter through complex 2 through FADH2. And you recall that this enzyme is also an enzyme found in the TCA cycle. So the electrons actually initiate from succinate that are then passed to FAD to generate FADH2. What happens to those electrons after they're passed to FAD? Let's take a look at that. This is complex 2 from pig heart. And as you can see again, the matrix is at the bottom of the slide in panel A, and the intermembrane space is at the top. Panel A shows a ribbon diagram with each subunit in a different color. And again, B and C show the actual electron carriers in this complex. Again, you do not have to memorize what these are. I just want to show this to you so that you can see that each of these, again, is a single electron carrier. So we're going to transport 1 electron at a time through this complex. And again, ubiquinone is going to pick up these electrons 1 at a time. And again, this complex does not pump protons, and it's eventually going to transfer its electrons to ubiquinone to make ubiquinol. The electrons from complex 1 and complex 2, and also from the glycerol 3-phosphate shuttle, can be used to reduce ubiquinone to ubiquinol. In this diagram, we see coenzyme Q, which is also known as coenzyme Q10. On the left, we see ubiquinone, which is a fully oxidized form of coenzyme Q10. This is a quinone with 2 double-bonded oxygens as substituents of the central ring. We can add 1 electron at a time to form semiquinone intermediates, shown in the diagram in the middle. When coenzyme Q is fully reduced, it is known as ubiquinol. This long hydrocarbon tail makes coenzyme Q, ubiquinone, ubiquinol, very hydrophobic, so that it can pass easily within the plane of the inner mitochondrial membrane to shuttle electrons between carriers in the electron transport chain. So which one is this, ubiquinone or ubiquinol? Can you tell? What should you look for? That's right, this is ubiquinol. Very good. Ubiquinol carries electrons from complexes 1 and 2 and from the glycerol 3-phosphate shuttle to complex 3. And this complex will pump protons from the matrix to the intermembrane space. Next, these electrons can be passed to cytochrome C. Here we see a ribbon diagram of complex 3. In panel A, we can see several subunits, each in a different color. In panel B, we can see that this figure also includes cytochrome C that is bound to complex 3 to accept electrons from complex 3. Panel C is transparent to show the electron carriers within these two proteins. As you can see, these include iron-sulfur clusters and also cytochromes, which are porphyrin rings. Cytochrome C has a porphyrin ring, and it's going to carry electrons one at a time from complex 3 to complex 4. And it does so by traveling in the aqueous environment of the intermembrane space. And so cytochrome C oxidizes complex 3 and reduces complex 4 by transferring electrons one at a time. Here we see their cartoon showing electrons being passed from cytochrome C to complex 4. Now, as these electrons go through complex 4, energy is given off that allows protons to be pumped from the matrix to the intermembrane space, and so complex 4 also pumps protons. This shows a ribbon diagram of complex 4, comprised of several subunits, each of which is colored differently in panel A. In panel B, again, we see a transparent structure showing where the prosthetic groups are in this complex that will actually do the transfer of electrons from cytochrome C to molecular oxygen, which is the final electron acceptor. Now, as you can see in this diagram, the prosthetic groups in this particular electron carrier, though, are unusual in that they include copper ions. Let's take a closer look at that. The role of complex 4 is to reduce molecular oxygen to water. And in order to do this, oxygen must not leave as a superoxide because that's dangerous. And so the structures that hold molecular oxygen in this complex are very important. Molecular oxygen is held as a peroxide bridge between an iron and a copper ion. The iron is located in a porphyrin ring, and the copper is held in place by several unusual amino acids, histidines that are bound to tyrosines. So what happens is 2 electrons and 2 protons are going to be added to this peroxide to form 2 hydroxyl groups. And then 2 more electrons and 2 protons are going to be added to these hydroxyls to make 2 molecules of water, which are then released. In this way, oxygen is safely reduced to water. And so this is the end of lecture 21. We've finished our discussion of the electron transport chain. We're going to talk about ATP synthesis in the next lecture. That's it. Hope you have a great day.